Hello everyone, welcome to the new session of Soil Management Part 3. In this session, we are going to study about causes of soil salinity. So what are the different causes of soil salinity? We are going to study about this. Soil testing, how to test the soil. Soil sampling, different methods to sample the soil. Soil test interpretation. So what we can interpret from this doing the testing of the soil, so that we are going to study in this, and also the field preparation and special practices related to soil. So these things we are going to cover up in this particular session. Okay, so the first one is the causes of soil salinity. Arid and the dry conditions. So this causes the soil salinity when the condition is quite arid or the dry, like the condition that are present in the desert, that is the arid and dry condition. So that causes the salinity. High water table. If the water table is high, then also it causes the salinity. Slopy land that washes out salt in the catchment area. So in the catchment area, sometimes there is a land are quite slope, right? So that washes out salt in the catchment area. All the salts along with the water get deposited in the catchment area, and all the salt that deposited in this catchment area. So that causes soil salinity. Irrigation with the saline water. When we irrigate our cropland with the saline water, then also it causes the soil salinity. And the poor drainage. So salt are present in the soil, but if there are poor drainage system, then the concentration of that increases, right? Reclamation method. The application of gypsum, that is the calcium sulfate, is effective for the improvement of alkaline or the sodic soil. So when we apply the gypsum that contain calcium sulfate, right, and it is an effective for the improvement of alkaline or the sodic soil. So that is the reason. It reacts with the exchangeable sodium present in the soil and convert it into the sodium sulfate. So what happened? Sodium is present in the soil, right? And it react with the exchangeable sodium present in the soil and convert it into the sodium sulfate. So calcium that is exchange with the sodium and it causes sodium sulfate. Sodium sulfate is leached out from the soil to reduce the soil pH. So this sodium sulfate is leached out from the soil and it reduces the soil pH also. Saline soil can be improved by ensuring the effective drainage system. So when there is an effective drainage system, that the soil salinity can be improved. So scrapping of the surface of salt from the highly saline patches is beneficial for the soil. So sometime what happen in the crop field, patches of salt can be seen. They are white patches, right? So we can scrap out those patches there, okay? And the soil, that part of the soil can be removed. So that is one of the way to minimize the salt content in the soil, right? So this is highly saline patches, and that removal of that is beneficial for the soil. Use of acidifying the fertilizer. So we can use the superphosphates and ammonium sulfate to minimize the salt content of the soil, right? So this is one of the way to minimize the salt content. Green manuring, that dhencha, sunham, mung bean, or the addition of the organic matter reduces the salinity of the soil. So some of the green manuring like dhencha, sunham, mung bean, and also the organic matter or the addition of some organic matter like the leaves, stem, branches, all that can be added, okay, as a green manure, and that also reduces the salinity of the soil. Soil testing. The soil testing help the ascertain that the status of the various nutrient, soil fertility level and pH, etc. To get ascertain that what is the status of the various nutrients that are present in the soil. We also able to know the soil fertility level. What is the soil fertility level, and also the pH of the soil. It is important to know the fertility status and the physical properties of the soil for maximum production and the rational soil management, right? So it is important to know what is the fertility of the soil, what is the physical properties of the soil for the maximum production and the rational soil management. A complete soil test program. So we are having a particular soil test program essentially consists of 3 basic steps. What are those 3 basic steps? Soil sampling. We can do soil sampling, soil testing. After the sampling, the next step is the soil testing and the soil testing interpretation. After that, we can interpret what is the type of thing that are present in the soil, like pH, the factors that are the pH, that is the soil content, right? So all these things we can interpret after the testing of the soil. So what is the purpose of soil testing? Why we do this testing? It helps to evaluate and improve the soil productivity. So it helps to improve the productivity of the soil. It helps to determine the nature of the soil, that is whether the soil is alkaline, saline or acidic. So this is also very, very important to know the nature of the soil. It helps to ascertain the use of fertilizer and manure and their doses in order to improve the fertility of the soil. So before adding any fertilizer to the soil, any manure to the soil, this is very important step to know the quality of the soil, and this can be done by doing soil testing. So once you know the quality of the soil, then only you can add the fertilizers according to that. Then only you can add the manures according to that, and to improve the fertility of the soil. So actually you want to know the doses that you are going to give to the soil of fertilizer and manure. And that doses can be decided after the testing of the soil. It reveals the actual condition of the soil so that it can be improved with the application of the nutrients and other management practices. Yes, so it reveals the condition. What is the condition of the soil, right? And it can be improved. How can it be improved? It can be improved by adding some nutrients in it, with the help of adding fertilizers and manures, and also some other management practices that can be followed to improve the soil fertility. Soil tests and their interpretations are based on the collection of soil samples and their analysis. So how to do this testing? So first one is taking soil samples and doing its analysis, right? So therefore the soil samples are taken represent the whole field. So there is such a big field. We can take sample from here, sample from here, sample from here and here, like this. And these samples actually denote the condition of whole field. It represents whole field, right? To obtain the information about the nutrient status of the soil, it is essential to follow the correct procedure of the soil sampling. So to obtain the information about the nutrient status, what is the nutrient status of the soil? Okay, so it is very, very important. And for that, we must go for the soil sampling. Before sampling information regarding the cropping pattern, various management practices, variations along with the direction of slope, soil color and texture must be noted down. So, what are the things before sampling? What we must note down? What is the cropping pattern? What is the management practices that we are currently following? Variations along with the direction of the slope. What is the direction of slope actually? What is the color of the soil and texture? This can be done prior doing the sampling. Then the field from where the sample is to be collected must be divided into different sections, and according to variation in slope and texture, all samples must be collected from each section. Means if there are two types of slopes, then samples must be from both the slopes. If there are three different texture, then sample must be taken from the three different texture. So that is very, very important before doing the sampling. By using the various sampling tools such as soil auger, soil tube, spade, etc., sample must be collected from the plow depth 15 centimeter normal agronomic crops and from deeper zone, that is 15 to 30 centimeter for the deeper rooted and the horticulture crop. So if we are having normal agronomic crops, then 15 centimeter depth of the soil that can be taken from the crop field, that is enough. And if we are going for the horticulture crop field, then 15 to 30 centimeter deep root. Okay, this is the depth of the sample that we take in the case of horticulture crop. This composite soil sampling is then spread on the clean sheet. If the sample is taken, then it must be spread on the clean sheet. It is divided into 4 equal parts and 2 opposite quarters are rejected and the samples from the 2 other are mixed. To obtain the desired size of the sample, 500 gram, then the same procedure is repeated. Before sending the sample to the laboratory, it must be dried and put into the plastic bag. So for the testing, we send it to the laboratory, and before sending it to the laboratory, the soil sample must be dried and it must be put in the plastic bags. The bags containing the sample must be labeled and sent to the nearest soil testing laboratory. So labeling is very, very important. Otherwise, it is of no use. You will never know that the sample belong to which field, right? So labeling is important, and along with an information sheet having the following details. So these details must be there, okay, along with the labeled sample, name and address of the farmer, identification mark and the number of the field. So there are actually the number of field. Every field has its number. So the number of the field should be there, and date of sampling, which date the sampling was done and the sample has been collected. Soil sampling, local name of the soil if any, color of the soil, that is dry and moist, type of land, unirrigated, irrigated or the waterland. So what type of land from which the sample has been collected, that should be noted. Source of irrigation, what is the source of irrigation to that particular land, canal, well or tank. Depth of sampling, this should be known. What is the depth of the sampling? And also the topography, level, slopey or undulated. What is the topography? That is at a level ground, it is a slopey ground or undulated, like this, undulated, slopey or level. So these all information must be there if we attached a sheet. Crop rotation followed. What type of crop rotation is followed? This should also be there. Previous crop, all the information that what was the previous crop that was grown in this particular field. Next crop to be taken and what is the plan, what we are going to plan for the next crop. Manure or the soil amendments applied earlier. So what were the manures or the soil amendments that has been applied earlier? So that should also be noted. Any other remark or any other information you want to give. The help of the village level extension work can be taken to collect the soil sample and fill in the information sheet. So we can take help from the worker that are working in the field or even the farmers to which the field belongs. So that from that the soil sample has been taken. So all these information must be there in the sample sheet. The collected sample is analyzed by using the standardized method in the laboratory for the following parameters. So what are the parameters? pH indicates whether the soil is acidic, alkaline or neutral in nature. So if the soil is acidic, the soil is alkaline or neutral, that can be analyzed by pH, knowing the pH of the soil. Presence of the total soluble salt. So what are the total soluble salt that are present? Okay, that is determined by EC, which indicate whether the soil possesses any constraint to the seed germination or the subsequent crop growth. So this is the presence of total soluble salt that are, if we analyze that, then we are knowing that what the soil possess, that is constrained to the seed, whether the seed's germination is affected or the subsequent crop growth is affected. So that we can know. Lime and gypsum if needed. So we can add lime and gypsum. After the analysis, we came out that either the lime or gypsum can be added if needed. Organic carbon, which is a measure of nitrogen content in the soil. Phosphorus content in the soil. Phosphorus content in the soil, that can also be added. And potassium content in the soil, that can also be added if required. So this, what is the requirement? Which of the following is the requirement? That can be only we know that when we are going for the soil testing. Rating the soil on the basis of EC. EC means electrical conductivity. Right? So soil is normal. It is normal when the electrical conductivity is below 1. So how to know the electrical conductivity? That can be known by estimating the electrolyte that is present in the soil, that is cation and anion. If we know the cation and anion, that is type of present, then we can know. Critical for the germination. If it is between 1 and 2, then it is critical for the germination. Critical salt level for the growth of the sensitive crop, that is between 2 to 4. And injurious to most crop, then it is greater than 4. So this is the rating of the soil on the basis of electrical conductivity. Okay, rating of the soil on the basis of the nutrient available. So what type of nutrient available in the soil? So that is organic carbon. If it is less than 0.5, then it is low. It is between 5 to 75%, then it is medium, and less than 0.75%, then it is high. Available nitrogen, actually this is 0.5, okay, and this is 0.75. Available nitrogen, that is less than 280 kilogram per hectare, then it is low nitrogen content. If it is between 280 to 560 kilogram per hectare, then it is medium, and if it is above 560 kilogram per hectare, then it is high. Available phosphorus, less than 10 kilogram per hectare, then it is low. Between 10 to 20 kilogram per hectare, then it is medium, and greater than 25 kilogram per hectare, then it is high. Available potassium, less than 110 kilogram per hectare, then it is low. 110 to 280 kilogram per hectare, then it is medium, and high, 280 kilogram per hectare, then it is high. So this is all about this session. We will have one more session to complete the soil management. Till then, thank you very much.